Recombinant DNA is DNA that contains DNA from more than one organism. And this is made by inserting a DNA fragment from one organism into the DNA of another organism. Usually this is of a different species. In order to make recombinant DNA, we have to use what's called a vector. And a vector is a tool that's used to insert a DNA fragment into a DNA strand. And there are two types of vectors. A mechanical vector, and an example would be like lab equipment, such as a pipette, and a biological vector. And an example of that would be like bacteria or viruses. The transferred gene will become part of the DNA, and that can then be put back into the original organism. And that's when you get a transgenic organism, or an organism that contains DNA from more than one species. We use recombinant DNA in many different areas. Here are the three main types: agriculture, medicine, and other industries. With agriculture, we use it to produce crops to make them last longer, taste better, to be resistant to herbicides, to produce biological pesticides. And an example of that would be inserting the Bt toxin gene from a bacteria into cotton. And when insects try to eat the cotton, they'll actually die because of that Bt toxin gene. With medicine, we use recombinant DNA in various vaccines, to make human growth hormone, insulin, which we'll talk about in detail, also with antibodies. And we also see this used in other industries such as food production, paper production, sewage treatment, or even production of various dyes. There are 5 main steps to making recombinant DNA. The first is to remove the gene that we're interested in by isolating it and using a restriction enzyme to cut that gene out. We then take a DNA plasmid, which is a circular piece of DNA found in bacteria, and we cut that plasmid with the same restriction enzyme that we isolated the gene with. That gene is then added to that circular piece of DNA, or plasmid, and now we have recombinant DNA, or DNA that contains DNA from another organism. That recombinant DNA can be put back into the bacterial cell, or put into another organism. If it's put into another organism, we get what's called a transgenic organism. And that transgenic organism is not a new species, it's just a genetically modified organism. In other words, it contains DNA from more than one organism. And that transgenic organism will then have the trait of whatever we were trying to isolate to begin with.